I tasked myself with talking about the future therapies and newer targets. One, because there's never been a greater time to look at what's available. Two, because we're taking information from other solid tumors and bringing it back to melanoma, whereas in the past it was all the work being done in melanoma and sent to other tumors. And three, I can say whatever I want, and no one's going to call me on it. But it's really nice. I titled it WTF, and that is, what's the future? Here are my financial interests and relationships and conflicts. But two things. One, the more conflicted, the better and more even keel the talk. And number two, if you look around, there are people from multiple pharmaceutical companies here just that should make you feel happy in that we do have a lot of basic science partners, surgical oncology partners, leadership in the hospital partners, but also now pharmaceutical partners who are really invested in the well-being of our patients and have a whole host of options. And this was not the same when we started 13 years ago talking about an AIM symposium. Here are the approved agents and what we have, and we've really moved average survival from 6 months to 6.5 years and more. And I feel, so what are tomorrow's questions that are due today? This is really what's going on. What do we do after the LAG-3, the relatlimab approval? What happens with CTLA-4? And then when you've exhausted Dr. Sullivan's options, what about TIL therapy? Are we done? Is it, what about brain metastases and leptomeningeal disease? We're seeing less and less patients have those issues, but when we see them now, they've exhausted all the options we've had. And then, of course, the whole reason we do this is the belief and buy-in into clinical trials. Dr. Sullivan talked about a trial that was started in 2013 and is reading out now. I would say that when we started with our initial immune checkpoint trials, it slowly seeped into the community. It took 10 years for people to even understand that we were doing trials with CTLA-4, but now you can start a trial and finish it and report it in a couple of years, 2 years. In the time that the patient who gets on the trial, 2 years in, there's data to help the whole cohort of people around. So it's fast, it's moving quick, but we need more answers, and it's not easy. This was what was, and Dr. Ferries has shown you this, but this is Jed Wolchuk's initial T-cell, blah, 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 blah. But in LA, as we saw driving around, you need a better vehicle and you need to do better. This is a electric car with lights and bells and whistles, and that's what I like. We're also learning that not only are we doing more, but we have a better goal. Our initial goals were, we just need to stop this thing in its tracks. And now we're getting people to this thing called CR, complete response, where everything is gone, or complete metabolic response, where you see things on the scan, but on the PET scan, they're not active. They're not metabolically active. And what these curves are telling us is that we need to work and build and do more and add more to get a greater proportion of patients to that CR, because those are the ones that are going to do the best long term. So we can't just sit on our laurels. And so I went to AACR. The slides at AACR would just happened in New Orleans. Unlike some of my colleagues who were, going down Mardi Gras and gathering beads, I looked at these things, and I think it's really worth mentioning that the ability to think beyond just one drug and another drug, and let's just give them together, is where we're going. Let's take one drug and make it like 5 drugs. And so This is a monovalent bispecific antibody. All it means is instead of just having PD-1, it has PD-1 and CTLA-4. And that allows it to target better. That allows us to get higher doses into the right places and less toxicity. So that's what this is saying. You can get faster inhibition, less toxicity. And that's been the problem with CTLA-4, the ipilimumab and the Yervoy you hear about. When you combine them, there's too much toxicity. But this type of bispecific drug can give you more. And then you can flip out and give LAG-3 instead of the CTLA-4. And these trials are ongoing in multiple solid tumors. This trial is a MedImmune drug, was in many, many cancers, and they saw responses in gastric, bladder, Merkel, cervical, and others. So these are coming, and they're coming for patients who have seen the standards in melanoma and need other options. And so many years ago, when I came to Angeles and talked about expanding the clinical trials department, I went in my office and there was no expansion to be had. There was nothing to buy. There weren't these trials, but now it's the exact opposite. We are looking and teasing out of tens of trials offered to find the best trials out there. Again, we're also looking at giving drug directly into tumor to try and avoid side effects, but to get it to start that tumor death that can then work systemically with the other drugs we give. And this is a CTLA-4 that's injected directly into the tumor, and you can see that it works distantly and locally. And this is in metastatic breast cancer. That's why I tell everyone to come to the melanoma talks, because you can take this back to other solid tumors and make breakthroughs. We have a lot of checkpoint inhibitors and checkpoint activators that are going into multiple clinical trials moving forward. And we have a lot to do here to understand the tumor microenvironment. But I'm just going to talk about one thing here, and these are adenosine inhibitors, partly because we have a trial of it here at Angeles. And the idea here is one that's super interesting. And no, we missed one. But it's that ATP is needed to give energy to our T cells to activate, go after the tumors, and to survive, to inhibit the bad T cells that put the brakes on your immune activity, to get the other immune cells into the area of the tumor, etc. And we know that this ATP adenosine axis is important, that when ATP is used up and becomes AMP, so ATP, you need it for your cells to grow and have energy, but the tumor preferentially takes ATP and breaks it down to AMP and then adenosine. And adenosine is inhibitory to our T cells. What we've found is that we can block CD73, which is an enzyme there, and we can block the target of the adenosine, and we can also block this other CD39 that changes ATP to AMP. Guess what? We're putting them all together in one trial. And that's the future, just not checkpoints, but other areas. When I hear people talk about neoadjuvant therapy, I think about all those patients who then come and say, I can't do that. I don't have tumor in place. But there's great stuff, great work that's coming out of multiple companies, including Moderna. You're all familiar with Moderna. And they're making vaccines off of the tumor that's already been pulled out of body. And in those patients who are getting adjuvant therapy without tumor in place, they make vaccines that express those proteins that can increase the immune activity. If you think back to the beginning of this meeting and Dr. Patel showing that increased T cell activity. If when the tumor is in place, you don't just need to have the tumor in place. You can have the tumor made into a vaccine given along with it. And guess what? That trial is already done, and that data will be presented shortly. We took a bunch of patients, a bunch being 150, and we gave 100 of them the adjuvant PD-1 inhibitor plus a vaccine made out of their tumor. And we gave 50 of them just the adjuvant PD-1 checkpoint inhibitor. And we were blinded to who got what. And that data is now 2 years in and will be ready to be presented shortly at a meeting by the end of the year. But you can identify these proteins that are the neoantigens. You can create a synthetic vaccine, and you can give it in combination with adjuvant and checkpoint blockade. You can also do it in the metastatic setting, and we've been doing those things, and that's coming forward. We then took a trip with Dr. Ferries and talked about TIL therapy. And that is, again, where the tumor tissue is procured. I don't have stick figures, but I have something between yours and Ferries' complex person. And so you take the tumor, you make these, you send it centrally, and they make these TILs and they send it back. Well, the idea is that we can do more here. And what can we do more? This is from a company that we're working with, and they are, I can't explain CRISPR, but CRISPR is a hot word. You say CRISPR and people think this doctor knows what he's talking about. He's at the tip of the spear in treatment. But so CRISPR, what they found is that they looked at these targets that lead to immune dysfunction, and they can CRISPR or cut out that part of it. So the place that did this, Iovance, is CRISPRing out PD-1, but this company is CRISPRing out SOC-S1. And this gene is inactivated, and you have an engineered TIL. So you have a heat-seeking missile that does not have a problem with it. It's stronger, and it goes forward. And what this says is, look, it does better than normal TIL. So you get no cells. This is how your tumor grows, this black thing here. You get normal TIL. This is how your tumor grows. And you get this drug. This is in mice models, and you get this. So when people say 10-fold potency, you want to put that in clinical trials, and it's coming. What about changing some of the drugs that give toxicity? How about giving away in order to avoid some of the toxicities of TIL? So Dr. Ferries talked about interleukin-2 and how long it can get rid of tumor, and how as an adjuvant, after you get those T cells infused back into your body, we'll go backwards here. You get those T cells infused, and then you get IL-2 in the hospital. How can we fix that? How can we make it better? And this is a drug that comes from a company named Obsidian, and they're doing their phase 1 trial at MD Anderson. And so they're going to start doing these trials at MD Anderson. They have, instead of systemically given IL-2, they're giving endogenous IL-15. That's another interleukin. We believe that it may be better, that it does not stimulate those Tregs, those cells that regulate your immune activity, and that you can see less systemic therapy from it. Anyway, there's a linker here, and you take a pill, and it activates that release of IL-15. So if you have someone, you don't have to give them these infusions. If they get toxicity, you stop giving them that pill that releases this. And you don't have to pay for the to. There's no financial toxicity of it. So it's more and more and more. And here are all of those scientific slides that say, hey, it may work better. Improved TIL product demonstrating anti-tumor potency without the need for IL-12. I'm up for that. Sign me up for that. And honestly, I came in second for number of slides per minute of talk. So I've tried to cut those out. But what I've cut out from this talk is the fact that TIL therapy is not just for melanoma anymore. There's great data in cervical cancer, and it will be in front of the FDA for approval for cervical cancer. There's great data in patients with lung cancer. There are trials going on with head and neck cancer, and it's moving earlier and earlier and earlier. And these TILs are going to come to patients and are already in clinical trials of patients who show and say, I have metastatic melanoma. I don't know if I should get PD-1, should I get TIL? And you go, guess what? You get both of those things. So that's upcoming now too. Why is it so interesting for us, and why are we pivoting our research program to try and bring some of these cellular therapies forward? It's not just because of this waterfall plot that Dr. Ferry's adeptly educated us about. It's also about these things. That's plots. Thank you. You have to learn about these plots. Way back when I was a fellow, you didn't need to do plots. I mean, nothing really worked. So they said it worked this. It had this response rate. It only worked for 2 months. So if they did like a swimmer's plot, it would stop like right there. You wouldn't need anything. But now you need to learn about the waterfall plots. That's how much shrinks. The swimmer's plots, very descriptive, that how long it works. And this is a very convincing response in patients who had seen, all of them, 100% had seen PD-1, 80% had seen anti-CTLA-4, which is Yervoy ipilimumab in one way or another. All of the BRAF mutant patients that Dr. Sullivan talked about had already seen BRAF inhibitors. And this worked. And it worked early. You see these blue diamonds? That's when the first imaging, a majority of them had already shown shrinkage. But here, 80% of the people that responded, their initial response to PD-1 therapy, which is the backbone, which is the standard therapy for every patient with metastatic melanoma who shows up in the door, 80% of these people, their best response to PD-1 therapy was no response. So that's taking a subgroup of our patients and telling them, we have something for you. If at 3 months when we look in, it didn't work for you, we have something for you. Next. Dr. Memi is going to come and talk about uveal melanoma, and I'm going to talk to you about how immunotherapy is now working in tumors that we never thought it could work. But this drug, you'll hear a lot more about. I'm going to say it's worked extremely well. I call it a matchmaker. Those of you who are Jewish like me, it's a yenta drug. It brings these two things together, but it's like a bizarro yenta. It puts you together and wants you to get a divorce. It brings the T cell and the cancer cell together and hopes that you get a huge immune reaction. And we're doing a whole host of trials with drugs like this that take other solid tumors like endometrial cancer and ovarian cancer, those that don't respond to immunotherapy and are seeing some promising results, right? During the pandemic, I learned to do a lot of animation on these slides. But this is the data of it of this drug in combination with PD-1 and CTLA-4 in cutaneous melanoma. So we are seeing about a 37% control of disease, and this trial is going forward. Guess what? It's being given subcutaneously under the skin. You don't even have to sit somewhere. You just go in, you get a subcutaneous injection of this drug. We watch you for an hour, kick you out. So it's great. The third type of plot you need to know is spider plot. So new things in your vocabulary is waterfall, swimmer, and spider plots. And this shows if you want the spider's legs to go downward and die like this, durable response. This shows patients who've had the standard therapies beginning to benefit. And we've taken patients off of this 2 years in and seen that they continue to have response. And that's the benefit of immune therapy. You don't have to continuously get it. Your immune system is active and going forward. Dr. Memi has brought forth other trials where we are working to utilize your own T cells, but we don't need to have you, come into the hospital, and we don't have to manipulate the T cells in a major way. The T cells come out, they're bathed in a jacuzzi of activators, and then they're given back to you in the clinic. So that's a wonderful future. You've heard about CAR T, but more and more this will come forth in other solid tumors and then come into melanoma. And this is the flipped paradigm. It didn't start in melanoma, but it's coming in melanoma. And then, I'm going to run out of time, but there are other types. And this word is a new word for you too, allogenic. That means it comes from someone else. And all of the CAR T stuff we talked about is auto. It comes from you, the patient. Allogenic is off the shelf T cell therapy. And that's NK cells that you can get off the shelf and give it to patients with other solid tumors. This stuff comes from Rizvani and others. Who is at MD Anderson? She's my favorite person at MD Anderson. Why? Because she's Iranian like me, and she's doing things like, we come to work and we're like, we're doing some great stuff. She's doing great stuff. She's taking cord blood, not even from an adult, cord blood, and utilizing it to make these NK cells to give to patients and manipulating those cells. So this is like a maestro. This was presented from MD Anderson Cancer Center. It's from her lab. And this is cord blood derived NK cells that have an innate engager. What does that mean? You don't need to remember that. It's basically a bispecific already hooked up to the NK cell. That's super. This was at AACR, which just could like jaw drop moment, because you don't give them together. You don't give one and give the other. This together, one drug that takes the NK cell directly to where you want it, to the tumor. And you can see this happening. We have that bispecific in melanoma. We have CAR TIL. We can do NK and go from there. What about brain mets? And this, I think, is towards the end. TIL therapy here. These slides I stole from Alison Betoff-Warner. This is data from the Iovance lifilocell that you saw in metastatic brain metastases, and she's going to be opening a trial like this. And so we're not just sitting on our laurels saying we have success, but we're tackling some of the difficult questions. Here's a patient who had systemic response and also brain mets response. Damn, I'm like an ad for MD Anderson today. Please tell him to send a check. Third floor. But, Isabella Glitza is phenomenal, and the whole MD Anderson history goes back to looking at leptomeningeal disease. That's the last bastion of how the worst outcome patients. There's a study Dr. Sullivan has of Ipi-Nivo and leptomeningeal disease. We know it works, but what if you've already seen it? This is intrathecal, giving it directly into the fluid in the brain. You don't go through the vein. You go, I'm going to put it in your brain. It's going to work, and it works. But there's better. This was presented from the Harvard group. It's not in melanoma. It's in low-grade gliomas, brain tumors in children. And they gave this drug. So this is CAR T. That's where Dr. Ferry said you change it. You give it something that hones the T cell to the tumor. This is CAR T, and they give it intravenously, that's IV, and they give it optionally into the cerebral ventricles, into the brain, a T cell. Just I'm going to put a needle into your brain. And what they found was amazing. These are children who had neurological compromise, who had resolution of all of their neurological symptoms. What they found is giving it intracerebral ventricularly without lymphodepletion, not needing to give that systemic chemo to get rid of the T cells, but just giving it into the brain. You saw activation of the immune system, and this was unbelievable that you're getting, this is your spine. This is the tumor. You don't want to see that. That's not great. But then shrinkage and shrinkage and shrinkage to near resolution. And the reason I use this pointer is because I want to bring your attention now to this. 2 months, 5 months, 9 months in to something that would have a very poor 1 to 2 month prognosis, if at best. So that's where we're going. And the next thing is to say that we are having better ideas of using imaging. So behind the cafeteria, we have MRIs, we have a PET scanner, and we need to learn how to use them. And this is an imaging tracer where you give it IV, and it hones directly to where the T cells are. So instead of saying, hey, you need to spend 3 months on this therapy, we can say within 8 weeks, we can do one early, see where your T cells are. And at 8 weeks, we can see if we're getting those T cells to your tumor. This is uptake in areas where there is tumor, and it's lighting up showing that the T cells are there. We overlay them, we say success. We overlay them and they're not on top of each other. We say, you need to go to something else. You don't need to come in another month or 2 months or 3 months. So with that, I am done.